as you collect yourself. Thank you all. Very interesting combination. So first we've got Vamsi, then Jeff Botkin.
No, I want to be there too. Yeah.
And Marnie, and then Keith, and then me. Okay, Vamsi.
So I have one question for Carlos, and then I have one question for, where's Doug? For Doug. So first, Carlos, obviously I think we had asked you to sort of talk about your tail-end work, but at the very end you actually alluded to the incompatibility issue as well. And of course you've actually published on this in the past, and I'm thinking about the very nice PNAS paper that you had published a few years ago, or maybe many years ago, where you sort of mixed and matched mtDNAs and nuclear DNAs from humans and other non-human primates. And there you did report that you could detect incompatibilities, but you had to go, there was some point in evolution at which that incompatibility was detectable. I guess my question is, what we just heard from Doug is that in mice, he can see some evidence of incompatibility, but it's not an oxygen consumption measurement. It's not a gross phenotype in a cell. Rather, it's a subtle behavioral phenotype. So I just want to try to reconcile the differences between what Doug just presented and what you had presented in that PNAS paper a few years ago, and whether you think that it's fundamentally an issue of different species versus the resolution of oxygen consumption measurements.
Right. I mean, just to frame for the audience. So the mitochondrial DNA codes for these 13 proteins that have to interact with a large number of proteins that are coded by nuclear DNA. So there is a compatibility issue. So what we did a few years back was to try in culture cells to get cells that had the nucleus from the human and try to put mitochondrial DNA from different primates. And we saw that we could put mitochondrial DNA from chimpanzee and gorilla, but by orangutan, they could not coexist anymore because the incompatibilities were such that they couldn't even exist. Then when we analyzed more carefully the cells that had human nucleus and mitochondrial DNA from chimpanzee and gorilla, they were sort of normal, but they had a complex 1 deficiency. So they had some incompatibilities. Now that's a good point that Vamsi raised, because besides these gross enzyme activities, mitochondrial respiratory chain might have more subtle functions that might be related to signaling and other things like that, and that might affect behavior and cognition. So that's a perfectly reasonable scenario. And then there's the issue of heteroplasmy. That's a new concept that it might be bad, even worse than anything else, and we don't understand exactly why that is.
So maybe then I'll follow up with the question that I have for Doug. You use the analogy of HLA matching for solid organs, for example. In your opinion, do we understand mtDNA, nuclear DNA, epistasis well enough? Is that science well enough developed so that you could determine whether or not a woman and a potential donor are a match or not? Because of course when you look at these sequences, you're going to pick some R-squared value and then say that's a same haplotype or not. So is that science, in your opinion, well enough developed at this point so that you could say reliably that it's a match or not?
Well, first of all, I do think we need more data on epistasis. There's some wonderful data from David Rand on Drosophila, where he's mixed mitochondrial and nuclear lineages in different strains, and again finds differences in their behavior, their strength, their function. So I think epistasis is important, very important. I also know that we don't understand epistasis or know what the key factors are. One of the things that we did a number of years ago was actually make hybrids within the same species with different nuclear versus mitochondrial combinations, and you can also see some subtle differences. The question is, which ones are good and which ones are bad? I'm saying there's no way we can know that now. That's why I'm saying, given that this couple is deciding to reproduce and they're going to use mitochondrial replacement therapy, why not try and get as close a match to the mitochondrial DNA as possible? And actually, it might be a terrible idea. That might be the wrong mitochondrial DNA for that nucleus, but at least we're not violating the decision they made as a couple. Whereas if we just took a random mitochondrial DNA, then in fact they haven't chosen their mate. And yes, it could well be that that mitochondrial DNA would indeed be better. I'm not saying it wouldn't, but it's something we couldn't possibly guess at the time. So it was just a matter of this is an expedient to avoid a possible concern.
But is there a procedure today that would allow you to find the closest possible match? I mean, is it simply the Hamming distance between the two mtDNA? I mean, is there a procedure for finding the closest?
Yeah, we have a very efficient way of sequencing mitochondrial DNA. We have 18,000 mitochondrial DNA sequences into a single phylogenetic tree, and so we could make the closest match. Now, would you get an exact match? No, but you might be off by 1 or 2 nucleotides. Many of them would be synonymous. So I think that you could do a pretty good job of matching, but I think whatever you do, it would be better than random. I do admit, though, however, that there's a big concern because, in fact, if the number of egg donors is limited, then just like HLA mapping, you may not have a matched donor, and then you're going to have to make an ethical decision about how far afield you're willing to go.
A comment? A couple of things. One thing is, I think, is that just a couple of caveats, is that all Shoubridge cell experiments are done on an aneuploid, highly aneuploid cancer cell background. So you have to keep that in perspective. None of them are diploid cells that anybody has done. All of the mouse experiments are done with inbred strains, all of whom have problems, and all of whom may have developed mutations. And I think that it's, if I understand biology correctly, that every generation, mitochondrial DNA is exposed to half the alleles that it's never seen before from dad. And so it strikes me that there's got to be pretty good compatibility between what mitochondrial DNAs are out there, because every generation there's a new half set of nuclear genes that come into play. And so I must say I don't quite get the compatibility part.
Then I'm going to have to counter. So using aneuploid cells, we can put in different mitochondrial DNAs, and what we find is it changes the epigenome. So the mitochondria is sending signals to the nucleus, and the nucleus is changing its gene expression. So again, mitochondrial DNA itself may be simple, but the nuclear cytoplasmic interaction might be much more sophisticated. So in human reproduction, exactly right, we put half of the genes for the mother, half of the genes to get to the father, so the mitochondria is interacting with a pseudo epigenome that it already recognizes. And that, I believe, can adapt the other chromosomes to fulfill that role. So I agree with you on many levels, Eric, but I think we have to be sensitive to the fact that there's more to it than just a simple reproductive random exchange.
I don't disagree with that, but your last comment, I would say there's really not much science on that so far. The converting of epigenome.
That in itself is a helpful conclusion, right? There's obviously disagreement among the people who know this the very best in the world. So that, to me, is an important takeaway from that exchange. Do you want to follow up?
If I could just make one quick comment in response to what Eric had just commented on. I mean, there is a notion of prezygotic selection. Some people also call this meiotic drive, where, you know, in a mouse, for example, if you have the T complex, that might influence which other chromosomes you inherit at the same time. And my understanding, and I think Doug just commented on this a while, is that we don't have any such examples of that in humans that involve the mtDNA, but in other organisms like mice, there is evidence of prezygotic selection, which could be relevant in the context of MRT.
You'd have to have a heteroplasmic population for that to work, right? Because if you have a single, if it's homoplasmic, there's no question of meiotic drive. At least as far as mitochondrial DNA is concerned, you're going to get what you got.
I guess it would actually be a really interesting scientific experiment to perform, and it may be the case that there is some small amount of data out there that would actually inform this. But for a given mtDNA, right, are all possible haplotypes possible? Right? So if you take, right.
It's like you're writing a grant proposal on the fly here, yeah.
Yeah, this is typical.
It's way too risky, so it won't be funded.
Jeff Botkin.
Well, thanks for these presentations, although I must admit to being having a struggle with the science here. So my question is going to be perhaps a little bit broader. Our task is to think about human clinical investigations. So I wonder if whether you have thoughts about what additional animal experiments might be essential prior to initiating any human studies in this domain.
Animals or cells? Would you include cells? Yes. Yeah. Okay. Okay. So for the cells, I think we need to do chromosomal spindle transfers using oocytes with a mitochondrial DNA mutation to confirm that not only that we can transfer the nuclear DNA with very little mitochondrial DNA, but then to take those cells and, if it's allowed, to differentiate them and make sure that they can differentiate into neurons or cardiomyocytes and maintain their mitochondrial integrity and show normal respiratory functions. So that would be one suggestion, because that's not been done yet. And similarly for pronuclear transfer. Yeah, I guess I agree with that. I don't think, for me, I mean, I'm not an expert in embryology, but for me, the transferring of the chromosomes on the spindle is probably the trickiest part of this whole procedure. And I wouldn't be very concerned about transferring, 1 or 2 or 3 or 4% of mitochondrial DNA from the mom-to-be's oocyte, because I think that's well below any kind of threshold and is very likely to get lost in subsequent generations. I'd be much more concerned about disturbing the chromosomes during the transfer procedure. But I agree that, I mean, the macaques that have been made, from what one reads, seem to be perfectly normal, and the procedure worked. But again, I don't know how many it took to get there either, because from my experience with manipulating mouse embryos, it's all a numbers game, and I don't know what the numbers are.
From what I've read, I think several of these embryos fail, but the ones that don't seem to develop. Now, I think more experiments would be good to have with embryos, maybe even human embryos, to see how good the efficiency is. But I think we're pretty close to whatever could be done with embryos is close to being done. I think we're close to, maybe trying to go all the way. That's how I see it.
Great. Marnie.
Thank you, of course, to all of you again. So I guess I have two questions. One, at the end, Doug, on your slide, you said that it would be best to use a maternal donor when possible, and I was surprised by that statement. So I was wondering if you could just explain, given the risk that they might also have disease. So that's my first question. And my second is an opinion of the panel on whether it's going to be important to test the donor in general. Maybe this should be done, for everybody, to find out their mitochondrial DNA genome to begin with, given what we know about the new, the incidence of mitochondrial DNA diseases in general. So I'd love to get your thoughts on that.
Yeah, you're absolutely right. If you are looking at a maternal lineage and let's say it's a sister, that would be probably very problematic because she would probably also be heteroplasmic but at a different level. What I was envisioning is we've looked at some very large pedigrees of things like the Milas mutation, the 3243, and if you look enough maternal crosses down upstream, you can sometimes find a case where, in fact, the maternal relative, it split off before that mutation occurred.
Sort of like a cousin.
Yeah, a
Got
cousin
it.
or second cousin or third cousin, yeah. So what was the second question again?
Just whether we should be doing anything more than just haplogroup matching, whether you should
yeah.
actually be screening the donor for mitochondrial DNA mutations, and if so, how?
Well, again, quoting Patrick Chinnery's group, he's looked at the I think it was the 10 most common pathogenic mitochondrial DNA mutations in cord blood, and he concluded that he could find one of those 10 in 1 in 200 cord bloods. So that means if you really tried to get a HLA-like panel of a large number of haplogroups, a significant let's see what that would be, 0.5% of all of the women would already have another pathogenic mutation. So yes, characterizing these mitochondrial DNAs, I think, would be a very important thing to do. How to do that, our technology is actually still fairly limited. I think Eric, or maybe I think Eric related to low levels of heteroplasmy, as people are finding by next-generation sequencing, where there are a lot of technological issues about whether those heteroplasmies are real or not. There are nuclear pseudogenes that come in. There's sequencing PCR errors. So finding a system where we can definitively say what a mitochondrial donor's mitochondrial DNA is like, that's another technological barrier.
And would you recommend doing it in the donor's blood? Would that be useful?
That is going to have to be a surrogate, because it's not going to be even reasonable to get women to donate eggs and then reject them. So we're going to have to do a pre-screen, probably as Carlos mentioned, of several easily obtainable tissues, hair follicles, blood, and so on, and hope that you can get a reasonable answer. But I'm going to let him talk to that.
No, I mean, just trying to find a compromise for this compatibility issue. I don't think in humans there is an evidence of this incompatibility between mitochondrial DNA from different races. We can discuss the epigenetics. That might be a gradual thing. But I think if you have a pool of donors and you can sequence the mitochondrial DNA and find a haplotype that's close enough, it's probably a good compromise that you don't have to get the same maternal lineage out thing. But yeah, you should sequence the mitochondria.
You're saying like don't just look for the haplogroup with a few markers. You're saying sequence the full genome, make sure there's no mutations that the mother carries asymptomatically. Right. I just want to make sure we're all in agreement.
I would.
Okay.
Well, yes.
I'm sorry. I wasn't clear. Yes, sequence the mitochondrial DNA. And in fact, we need to sequence the mitochondrial DNA by the best current methodology, because there are a number of papers in literature that are themselves, I think, ambiguous. But we can sequence a couple of hundred mitochondrial DNAs a month now, so I don't think it's a matter of a difficulty of getting the sequences.
Yeah, and I think I would add urine as one of the tissues to sample, because as Eric Shoubridge and we have observed, often these pathogenic mutations are higher levels of heteroplasmy and more easily detected in urine than in blood.
It's just would you screen for mitochondrial DNA deletions too, and if so, how?
Well, I think next generation sequencing often picks up the deletions at low, yes, big ones, yes.
So Keith, you're next on the list, and it's me, Jonathan, Alta. Anyone else? Okay. Okay.
Yeah, I'm not sure this is a question, but a comment that I'd like you to respond to, which is that I think the presentation, we might have started early on with the statement, anything we can do to make this machine, this mtDNA machine, work better is something we should do and we owe to families. But I think by the end, Dr. Wallace, anyway, raised the kind of a question, that is to say that what comes to light is that there's sort of a good internal discussion, there not being a consensus on whether MRT would necessarily make this machine work better, and a kind of a clear-eyed understanding of what the trade-offs are. So in one trade-off is no MELAS mutation, but mild cognitive deficits, which is something I came away, which I think many of us might say, well, that's actually a reasonable trade-off. But there's some other trade-offs that you're highlighting that are maybe problematic based on what we know from the studies so far. So I wondered if the committee, if the panel can talk a little bit more about what the evidence is from your world about these trade-offs, which is, I think, a different way to think about this technology, not a technology that's all good or all bad, but in which there will be some trade-offs. Am I reading the temper of the conversation clearly, or am I misreading it somehow?
Yeah, I'm sorry I wasn't clear. I was not implying that there would always be a negative outcome for mitochondrial replacement therapy. I was saying if we had closely matched mitochondrial DNA and there is not any unexpected change that could, say, alteration of the structure of the cytoplasm, let's say, or some epigenomic change. But from the genetic point of view, I think if you could substitute a very similar mitochondrial DNA, that should be a good outcome. That would not necessarily have any negative effects. So I don't want to imply that I thought all of this was a risk genetically. Okay.
Thanks for the clarification.
I agree. I mean, I think getting rid of the MELAS mutations is a no-brainer. I mean, the kids are going to be well, and I wouldn't worry about the cognitive things. And I think, to get back to the compatibility issue here, I think there are probably hundreds of thousands or millions of people who got married and had children who have different nuclear backgrounds and different mitochondrial DNA haplogroups. And maybe somebody should gather that data on human populations and simply ask the question, is there any statistical evidence for any kind of adverse outcome when you mix and match? Rather than relying on, Doug may be right, but I am a scientist. I'm skeptical, and I think there's a lot of pedigrees, like Carlos showed, where you can mix and match, and I'm not sure that it makes any difference in real human populations. But I think we should maybe gather the data, and there are children who have been made who are heteroplasmic, and nobody's mentioned those, the follow-up on the few that were made in 20 years ago by mixing cytoplasms, and I wonder if anybody wants to comment on that at some point.
So my question is actually a kind of version of Keith's, and to follow on to this. So the question's sort of if this were to go forward in humans, we'd want to be able to measure some of the kinds of sounds like subtle effects that you're talking about. And the data from mice that you showed, Doug, are, there's sort of obvious differences in those that are heteroplasmic from those that are not. But so the question is whether that's a function of there being highly inbred animal models, or whether that's something that might be detectable were it to be evident in a human if this were to go forward. So maybe you can talk about whether there's a kind of translation from the animal work into human, kind of a version of Jeff Botkin's question about what more animal data do we want, but going into humans, what do we want to know, and how are we going to see these subtle changes? Can we expect to detect them?
Well, the reason we use inbred mice is so that we can have uniform experiments, and therefore we have low variance so we can detect differences. Human population is by its very nature outbred and therefore highly heterogeneous. It might in fact be almost impossible to decide on a human experiment what would be the effect of such a subtle incompatibility. But I'm a clinical geneticist. I see all the people with attention deficit disorder and with schizophrenia and with bipolar, and what causes those? They look perfectly normal. They seem like anatomically normal human beings, but they're not happy. They have learning defects. So why is that? I don't know. I'm just saying I don't know that we could do that experiment in humans. I do know that in the outbred experiments with Drosophila, they do see a difference using wild strains. And also I understand that there have been some experiments on wild-type captured mice, but I haven't seen those published. But right now, you're absolutely right. There's so much variability in the human population, it's going to be very difficult to know whether that's a fact.
And maybe that's the buffer.
Well, right. I mean, there's sort of multiple ways to understand that. And okay. We'll talk more. So I have Jonathan Kimelman sending a question to Dr. Wallace. The last slide contained helpful suggestions regarding eligibility for applying spindle transfer to initial cohorts of patients. Any reason why this slide spoke strictly to spindle transfer as against polar body or pronuclear transfer?
I'm sorry, just repeat it again.
Sure.
Because I was hassling.
That was the first time I've read it too, so it'll help everybody, I bet. So the last slide you showed contained helpful suggestions regarding eligibility for applying spindle transfer in initial cohorts of patients. Is there any reason why to apply that slide spoke strictly to spindle transfer as against polar body or pronuclear transfer?
No.
That's really small print.
No.
Okay.
I listed spindle transfer only because the comparative data up till now shows that it has the minimum of the two nuclear transfer approaches, the minimum carryover of the donor mitochondrial DNA. But all those other techniques are valid.
Okay, thanks. So I've got Alta and then Marnie.
Okay, two questions. The first one's more important, I think. There was the comment that the mitochondrial variations might actually have been an adaptation to new environments during, population movements. And I was wondering what the evidence was that it was an adaptation as opposed to simply a founder effect, which would seem to be even stronger for mitochondria than it would be for nuclear DNA founder effect kind of analyses in terms of evolutionary theory. The second, I'm not sure what exactly I'm asking, except all the stuff about the mix and match. There's a long history, a really ugly history about race and assumptions about whether or not people can in fact interbreed, whether we're different breeds of people, et cetera. And I keep worrying about the way we're talking about this because it seems awfully close to kind of reinstating older versions of what race means and even adding a whole new kind of biological layer to it. And I just would love some reassurance, if there's time, that that's not exactly where this is going. But the more important question, I think, was really the evolutionary theory question. I just didn't understand that.
So the question here goes back to population genetic theory. This goes back to a man named Kimura, who in the 60s wrote a paper on how population allele frequencies change in derivative populations. And he was thinking about nuclear genetic variation. And he was a mathematician and wanted to understand population genetics, and he made an assumption. And his assumption was that deleterious mutations would be removed by what's known as purifying selection. And therefore, most mutations would be neutral. So using that assumption, he did a fabulous job on developing a whole new statistical mathematics of population genetics. No problem with that. And the most important aspect of that is what we call effective population size. That is, drift, or the frequency of a particular allele, will change faster if your sample size is small. And we know that. If you sample two balls out of a big bottle of black and white balls, you're going to get a very different statistics than if you sampled 500. Okay, so no question about that. And the mitochondrial DNA is maternally inherited. Therefore, instead of having 4 copies of nuclear genes, 2 from each parent, you're getting 1 copy from the mother. No question about that. So what's the alternative data? We've done a lot of work on the biochemistry of these mitochondrial different lines. Didn't have time to talk about it, but we studied Tibetan mitochondrial DNAs. There's a specific variant that has arisen in Tibet. It goes up in frequency with altitude. The odds ratio of that occurring by chance is 25. 3 independent events of that mutation on what we call haplogroup M, but never on N. And that mitochondrial DNA lineage on the M background gives the highest mitochondrial function. So that's just one of many examples. So we've spent many years trying to prove that these mitochondrial lineages are not neutral. Now, even today, my papers are argued with the neutralist theory that all this occurred by chance. And, I can't say that null hypothesis could ever be completely disproven, but an odds ratio of 25 to me is pretty convincing, and that we have quite a few examples of that. In relation to race, I actually don't feel I'm a racist. I am a human geneticist, and I think that you can find Caucasian genetic variation that's incompatible with each other. So I'm only saying there might be some genetic combinations that are incompatible, and we see that in the clinic all the time. There are people that are sick, but they're of the same race. So to just say that genetic variation is racist, I don't think that's an appropriate approach.
I'm not trying to say that at all. I'm trying to figure out a way to talk about this without it sliding into something that is not intended but could be very, very problematic.
Well, I put that slide to illustrate this fact that in human population there is no evidence that having the mitochondrial DNA haplotype from one race or one group and the nuclear from the other will have any deleterious effect. There's no evidence of that.
I agree.
So I think I'm next. So that's the perfect
You are
segue
next.
to my question. I'm so attached. So nobody brought up today, the effect of mutations, of course, and environment, right? And so there has been some theory, and I would just like to hear some thoughts on it, if we can, about somebody with a specific genetic background, be it mitochondrial DNA and otherwise, that grew up in one background that goes to a different background or a different lifestyle and gets complex diseases, things like diabetes, or obesity based upon nutrition or activity levels. And so I wonder if anybody up there can comment on that aspect of haplogroup, so not necessarily, inherited Mendelian diseases per se, right? But in terms of the complex interactions, I think that would be informative if we could just hear some thoughts on that.
I don't think there's any data on that. In terms of mitochondrial DNA haplogroups, people moving, say, from Japan to the United States and developing metabolic syndrome, which is unheard of in Japan, that sort of thing. But I don't think, but I mean, there's lots of examples of that going all different which ways of diseases, complex genetic diseases like metabolic disease or something like this becoming more prevalent based on diet. But I don't think, to my knowledge anyway, nobody's looked at that in the context of mitochondrial DNA haplogroups. Maybe Doug has other data.
There are some modifiers in the nuclear DNA. So if you have some mitochondrial DNA mutation that might confer hearing loss if you take an antibiotic, there might be some nuclear DNA that has another mutation that's mild that might compound that. So there are examples of that, but they're all sort of pathogenic mutations, not normal variations from what I know.
Sorry, I have to disagree with my colleagues again. So we've spent a great deal of time looking at mitochondrial DNA lineages and comparing them with predisposition to a wide variety of different clinical syndromes. And I think they're all highly statistically significant. I gave you a rough list of some of the things we found. I'll give you an example of macular degeneration, very common adult onset disease. There's a mitochondrial DNA lineage called U5. If you have inherited that from your mother, you have a probability of 1 of getting macular degeneration. So that clearly indicates that that has an effect on a predisposition to a disease. Whether an African mitochondrial DNA coming to the United States has a different predisposition to a set of diseases or not, these are things that are being looked at. There are several issues there. One, of course, there is the change of the, there is the introduction of the different genotype, but there's also the effect of the environment on that genotype and how that person adapts to it. So I think it is difficult to say something as broad as that, but I don't agree that there is no evidence that mitochondrial DNA haplogroups are genetically neutral.
Anyone else? Keith.
I just had a question, given the committee's charge. In thinking about the social implications of the committee's work, do you, is there, in your expert opinion, is there a good likelihood of limiting MRT to really clearly circumscribed groups of disorders, like MELAS, let's say? Or is, in effect, approval of the technique opening it up for a wide use of what you might call suspected mitochondrial diseases of a wide range? In other words, I'm trying to get a sense of what the scope of disorders that would possibly be affected by, because we've now talked about things like athleticism, energy, et cetera, and one of the questions before us is this line between disease and enhancement. So I wondered if the committee has a sense of whether there are Dr. Wallace is giving us some mechanisms by which we might think about limiting and circumscribing the risks, and I just wondered if you had some closing remarks with regard to this question.
I wouldn't touch enhancement, personally. I think it should be used for disease, and I think Doug Turnbull will be here tomorrow, won't he? And he and the Newcastle group have actually calculated, based on epidemiological data in the UK and in the United States, how many women who are carrying these disease genes might benefit from this technology. So he actually has the numbers for that.
I agree, and I think there's one of the last slides was pretty good on that. This should be restricted for women that have demonstrated pathogenic mutations that are known to cause severe diseases, and that's it. And I don't think it's going to be, by allowing that, I don't think we're going to be opening a gate to a big slippery slope. We can restrict it to that, I believe.
Yeah, and I agree absolutely with Eric and Carlos. And the number that Doug Turnbull published in the New England Journal at the end of February is about 780 women in the United States.
So I think that is a nice way for us to end today. We're breaking in the midst of the session on science and medicine, so we'll come back tomorrow at 8:30 and pick up. So keep the wheels turning. Thank you all for your comments and your participation on the panel. Very, very helpful. Thank everybody for being so compliant on time and schedule and making such pointed and well thought through comments. So we appreciate that very much. I'll speak on behalf of the committee. So good night. We'll be back at 8:30 in the morning and look forward to picking it up there.